Bismillahirrahmanirrahim. Our topic for today is gram-positive rods, divided into spore forming and non-spore forming. The spore forming includes the Bacillus and Clostridium species, whereas the non-spore forming include Corynebacterium, diphtheria, and Listeria monocytogenes. Another differentiating features between this organism is that the spore forming ones are comparatively longer and more deeply staining, whereas non-spore forming organisms are club-shaped and comparatively smaller and comparatively lighter staining, but they characteristically appear V-shaped or L-shaped. Now, another differentiating feature between these organisms is that all are aerobic. That means they need oxygen to grow, except for Clostridium species, meaning that they can grow in even dead tissues. Now, the last differentiating features between all of these organisms combined together is that all of these produce exotoxins, like Bacillus will produce it, Corynebacterium, Clostridium, except for Listeria monocytogen, which does not produce any exotoxin. It consists upon 4 species: Clostridium tetani, which causes tetanus; Clostridium botulinum, which causes flaccid paralysis; Clostridium perfringens, which causes gas gangrene; and Clostridium difficile, which causes gastrointestinal upset when you take a lot of antibiotics. As you know, this is an anaerobe, meaning that it does not require any oxygen for its growth, and along that it is spore forming. A topic is Clostridium perfringens causes 2 distinct diseases depending upon the route of entry. So if the route of entry is a wound, it would result in a disease known as gas gangrene. And if the portal of entry is by any food acquiring route, so it will result in food poisoning. Let's be discussing gas gangrene. Clostridium perfringens has a normal flora. It is found in both the colon as well as it can also be found in the vagina. Okay, so the transmission of this organism is that the soil contains this organism, which contains its numerous amount of spores. So these spores that are lying in the soil enter the human body when they suffer from any abrasion or any injury. So in order to cause injury, the most common things are that there are automobile accidents. It is more also seen in war wounds, and it is also seen in even septic abortions. So whenever this organism grows and enters through a traumatized area, it begins to produce a lot of toxins in that respected dead tissue. So that will be depicted such as, considering this to be the foot, and these are a lot toes, okay, and the person has suffered from an abrasion over here. Now here the organism will begin its growth in that tissue. Because it's an anaerobic organism, it will form a lot of toxins. Now the most prime toxin in Clostridium perfringens is the alpha toxin. Now, the function of this alpha toxin is that it enters from the dead tissue into the blood. Now, in blood, what it causes is, considering this to be the blood, a blood vessel, and that contains numerous amount of erythrocytes, or as you clearly known as RBCs. Now, this toxin enters the blood and destroys the membrane of these toxins, making it more porous. If it is more porous, obviously it will be destroyed, and a lot of hemocysteine or a lot of component within those RBCs would easily leak out. Such breakage of RBCs is known as hemolysis, depicted by a picture that in this dead tissue, a lot of alpha toxin is secreted that enters the blood, and similarly it destroys the RBC, resulting in hemolysis. Now, when this homeostasis is broken up, a lot of fluid would indefinitely leak into the respective tissue, and there will be a lot of stasis and a lot of hemocidin concentration, because that is the degenerative product of hemoglobin. This would give a characteristic yellow jaundice like appearance in the surrounding tissue, as well as there will be edema due to the leakage of fluid, so this leg would be increasingly swollen. In the clinical finding, we'll see that the respective area is very painful. There will be a lot of edema. As you can see, there is a lot of swelling of the leg. That tells us about the edema. There will be cellulitis that is present over this respective area. And whenever you press such a leg, you will hear crackling sound. These are actually crepitations because of the because of the gas produced in the respective tissue because of the proteolytic and degenerative enzyme secreted in that tissue. Also, this would give a characteristic amount of a pungent amount of smell because of the organism's growth. So the tissue next day we can see gram-positive rod. But normally what we do is we take we see the colonies of this organism on blood agar. On blood agar, what we see is double zone of hemolysis around the RBCs, which is a characteristic finding on C. perfringens, and on egg yolk agar we can see the alpha toxin, which is also known as the listeniase, which demonstrates the presence of this toxin being present there. Another picking up feature is that it can also do sugar fermentation and organic acid production, and the spores are not usually seen because they, as you know, already grow in a nutritional deficient condition. The treatment of choice for this organism is penicillin G. Prevention against such wound should be that wound should be adequately cleansed as well as debrived in order to remove a lot of dead tissue. By removing such dead tissue, the spores would not germinate or the organism would have no place to grow. Is that how Clostridium perfringens cause food poisoning? The portal of entry for Clostridium perfringens to actually cause food poisoning is quite simple. You engulf the bacteria. The bacteria will enter into the stomach and the small intestine. Now, as I've previously explained, this organism is already the normal flora of this organism is the colon, so it will not cause any specific disease in that respective area. But if this organism is present in the small intestine, upon engulfing, this bacteria would release endotoxin that would result in stimulation of the enterocytes. Enterocyte would be stimulated in such a way that result in increase in cyclic AMP, which would result in a lot of release of fluid from within the enterocytes into the gastrointestinal tract, resulting in diarrhea. The finding of this diarrhea is that this diarrhea has an incubation period of 8 to 16 hours. It manifests as watery diarrhea and a lot of abdominal cramps. And this feeling of abdominal cramps and vomiting resolves within 24 hours. This organisms are usually not done because the toxin cannot be isolated, and a large number of organisms can be isolated from uneaten food. So, no lab diagnosis. Because this disease is short-lived and symptoms resolve within 24 hours, so treatment is always symptomatic. Similarly, if in such a patient hydration needs to be corrected because of diarrhea, a lot of fluid is being lost, and for cramps, surgeons antispasmodic and antiemetics should be given to control vomiting.